أعزائنا الطلبة، أهلا وسهلا ومرحبا بكم في محاضرة جديدة من محاضرات الميتابوليزم. This is session 2 lecture 1. I am د.احمد جعفر. I will present the lecture of cell metabolism, bioenergetic and energy balance. In this lecture we try to understand the following learning objective: to describe the origin and the fate of cell nutrient, to define cell metabolism and explain its function, and to describe the relationship between catabolism and anabolism, and to explain the role of redox reaction and H carrier molecule in the metabolism, to explain why cell need continuous supply of energy, and to explain the role of high and low energy signals in the regulation of metabolism, and to explain the biological role of ATP, creatine phosphate, and other molecules containing high energy of hydrolysis phosphate groups. Chemical substances in the body, it's either cell nutrient, which include glucose, amino acid, and fatty acid, or waste products, which include CO2, urea, and NH3. Changes in the cell nutrient and their waste product, either physiological changes, which include after meal, during fasting, starvation, exercise, pregnancy, and during stressful condition, or pathological changes, and these are include in different diseases such as diabetes, atherosclerosis, obesity, shock state, malnutrition, and certain enzyme deficiency. All these conditions will cause pathological changes in the cell nutrient or waste products. Where is the origin and what is the fate of cell nutrient? Cell nutrients circulate in the body, come from variety of sources. These sources is either from the diet or from synthesis inside the body. Some nutrients are synthesized inside the body, so the body doesn't need for this nutrient, are so-called non-essential compound. Other route of origin of cell nutrient is the release from storage in the body. Some nutrient may be stored in the body, and when the body need them, there will be a release from storage sites inside the body. So these are the sources and of the cell nutrient. What is the fate of this cell nutrient? Cell nutrient either degradation. Either there will be a degradation to release energy into the tissue, or there will be synthesis of the cell component. This material, this nutrient, may be used for the synthesis of some cellular component. These cell nutrients may be used to be stored in the liver, or in the adipose tissue, or in the skeletal muscle. Some cell nutrient may face the interconverter ion into another nutrient, such as in the liver, adipose tissue, kidney, or the cortex. There will be interconversion. Or some of the nutrients may be excreted through the excretory organs, such as the liver, kidney, and the lung. So these are the fates of cell nutrient inside the body. So what is cell metabolism? It is a highly integrated network of chemical reactions that occur within the cell, and the network consists of number of distinct chemical pathways that's called metabolic pathways, which links to each other. What is the functions? What for cell nutrient? Cell metabolism? It's either for the production of energy or for synthesis of building block for building block of molecules, or it is the source of organic precursor molecules such as the acetyl-CoA, or the metabolism may be used for biosynthesis of reducing power like the NADPH, nicotine adenine dinucleotide phosphate. The metabolism either of two types, either catabolism or anabolism. The catabolic pathway is the breakdown of large molecules into small ones. It is oxidative, release large amount of free energy. Some of these energy conserve as ATP. Or the catabolism may be an anabolic pathway or anabolic reaction in which small molecules are built up into larger one. And these reaction and these pathway are reductive and use the intermediate metabolite and energy in form of ATP to derive the synthesis of important cell components. And we can see that in this diagram the metabolism and interconnection between catabolism and anabolism. These are the nutrient when enter the cell. It will be catabolized to smaller molecule. During this process there will be a release of energy as a heat, or stored, the energy stored, or sorry, it is carried out as ATP. And this process, when there is a catabolism of the nutrient, there will be precursor will develop from this process. This precursor will enter into anabolic pathways, and during this anabolic pathways there will be consumption of energy in form ATP, which converted to ADP. And during this process there will be a building up of large building blocks, and these large building block or macromolecule may be used for cellular structure, for cellular processes, or may stored as an energy storage substances. And these energy stored substances may be used again during the catabolism when the body need them. And during this catabolism some of the ADP may be converted to ATP. And this is the process of interconnection between the catabolism and anabolism, and turning out of the energy from one process to other process. The second concept is the biological oxidation. The oxidation is a combination of substances with O2, or loss of hydrogen or loss of electrons. The corresponding reverse processes is called reduction. العملية المقابلة أو المكافئة للأكسيدation, it is the reduction process. The biological oxidation are catalyzed by specific enzyme, cofactor, or coenzymes. And this is the process of interconnection and combination of both oxidation and reduction. When there is a molecule A, which is a reduced form, it will be oxidized to, when A to H, oxidized to A molecule and lost its hydrogen atom. This hydrogen atom used by another molecule which converted from its oxidized form to its reduced form by gaining the hydrogen atom developed or released during the oxidation process. So the redox reaction, which is the combination of reduction and oxidation, it is a a combination process, or it is a combination reaction, and it's considered as a one reaction when there is a release of hydrogen atom or release of electron from one reaction to be used by another reaction to convert the oxidized form of one molecule to reduced form of another molecule for the same molecule. And these hydrogen atom or electrons used by the another reaction, which is convert the oxidized form to reduced form. And this process of oxidation reduction are facilitated by some important cofactors that used for carrying the electron between the reactions. These factors are NAD, NADP, and FAD. Nicotine, adenine, and nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide phosphate, flavine adenine dinucleotide. These are complex molecules which contain components that cannot be synthesized in the body and have to be supplied in diet, such as vitamins. So these are an essential component of the nutrition of the body, which is the macronutrient. These macronutrients, such as the flavine, which come from the riboflavin, which is a vitamin, or the niacin vitamin, nicotinamide, come from the niacin, which is also one of the B class of vitamins. And these are act as a carrier for the electrons in these reactions, such as NAD plus 2H converted to NADH and release one hydrogen atom, or FAD with two hydrogen atom converted to FADH2. And this is the reduced form. This is the oxidized form, and this is the reduced form. It will be retained and converted to its oxidized form when it passes in an oxidation reaction. In this diagram we can see, and to continue the process of oxidation reduction, when there is molecule A, 2H converted to A molecule, it is released the hydrogen atom in an oxidation reaction to such as NAD converted to NADH from its oxidized form to its reduced form by gaining one hydrogen atom from this oxidation reaction from the other side. And this is process, the NADH plus H will convert it then also convert it to its oxidized form by releasing its hydrogen atom in an oxidation reaction to release the hydrogen atom which can use in an oxidation reaction in the other side, in which the hydrogen atom used from this compound, it is carry the hydrogen atom from one side to another side to facilitate the oxidation and reduction reaction. The reaction that serve to oxidize the reduced carrier molecule. When this carrier molecule is oxidized, what are the reactions? What are the processes that used to return back this molecule from its oxidized form to its reduced form? This reaction either during cell respiration, or it is called the electron transport, or during reaction in which substrate is reduced, such as the in the process of conversion of pyruvic acid to lactic acid, or it can be or this molecule converted from oxidized to reduced form during the biosynthesis reaction involve reduction steps, such as during the fatty acid and cholesterol synthesis require NADPH. During this, there is a biosynthetic reduction power. So this molecule, when it oxidized, it should be returned back to its reduced form, and this can be facilitated by one of these reaction inside the body. So the third concept, which is the bioenergetic. Bioenergetic is a quantitative study of the energy transduction that occur in a living cell, and of the nature and function of the chemical processes underlying these transduction. So when there is a transduction of energy from different molecules inside the body, there will be a release of energy, a consumption of energy. The science that study these processes, this is the bioenergetic science. Some concept we should understand in this study, which is the bioenergetic. We should know what is meant by free energy, or G, or Gibbs free energy. It is expressed the amount of energy capable of doing work during a reaction at constant temperature and pressure, and this could be the change in the free energy, the delta G, either below zero, which is in the minus, or and so the energy is exergonic reaction, when there is falling down of the free energy from high level to low level. The reactant which have a high energy capacity at the start of the reaction to become a low energy substance, or at the end of the reaction, when the reactant have high energy and the product have low energy, the delta G will be in minus, and this is an exergonic reaction due to release energy during this process. So the product energy minus the reactant energy will be in the minus. The reaction is exergonic, and the reaction is said to be spontaneous because it does not need energy to be stimulated. It is spontaneously occur, and during this process there will be release of energy. On the other side, when the product of the when the reactant have low energy capacity and end with a product which contain high energy capacity, so there will be energy added to the reaction, and when we calculate the delta G, it will be in minus, because the product energy capacity is higher than the energy capacity of the reactant, and the delta G will be the product minus the reactant energy will be delta G in the positive, and it is more than zero. And this is type of energy is said to be endergonic reaction, because and this reaction is need energy to be added to stimulate to start the reaction, and this reaction is said to be non-spontaneous reaction. The other concept is the enthalpy. Enthalpy is the heat content of the reaction it reflects the number and the kind of chemical bonds in the reactant. It is either exothermic when there is release of the energy, or endothermic when their reaction consume the energy, and the energy is conserved, is a chemical reactant, is the chemical in the chemical reaction. It is the energy is conserved in the chemical reaction, and the amount of the energy in the universe at the end of the chemical reaction is the same before the reaction takes place. The energy is the same, but it is conserved and stored in a chemical reaction. The other concept we should know in the bioenergetic is the entropy. And the entropy is the quantitative expression of the randomness of the disorder in the system. When the product of the reaction are less complex and more disorder than the reactant, the reaction is said to proceed with a gaining entropy. So the entropy is the randomness of the reaction. When there is an increase in the randomness of the reaction is said to be a gaining entropy. When there is decreasing in the randomness of the reaction and the product is more complex, said to be it is loss of its entropy. Under the condition existing in biological system, including constant temperature and pressure, changes in free energy, enthalpy, and the entropy are related to each other quantitatively by the reaction delta G equal to delta H minus T temperature by delta S. This can be summarized by this diagram. The delta G equal to delta H minus T by delta T, which is stand for the temperature, minus delta S. In the catabolism, as we said, the catabolism is a reductive process. It is a conversion of the molecule from large molecule to low molecule, in which the product will release energy. So the delta G, because of release of energy, the process is spontaneous, so the delta G will be in the minus. It is a release of free energy, and the delta G is in the minus, and the reaction is said to be exergonic, or releasing energy, the reaction that is releasing energy. While the delta H, it is because there is release of energy and heat, the delta H in the catabolism will be also in the minus. There will be release of the energy, so the delta H is in the minus, and the reaction is said to be exothermic. There will be release of the heat. While the entropy, because of the molecule in the catabolism converted from more complex molecule to less complex, and there is a destruction of the molecule, there is an increase in the randomness of the molecules, the entropy will be in the plus. On the other side, the anabolism, the delta G, there will be a gain of the energy. The reaction is not spontaneous, and it is an endergonic reaction and need energy to start the process of anabolism. The delta H, the reaction in the anabolism, it need energy, take up energy, so the delta H also positive, and the reaction is said to be endothermic. While in the anabolism there is a building up of molecule, large molecule from small molecule, so there will be decreasing in the entropy. and the entropy will be in the minus. The role of ATP and the ADP. The ATP, which is adenosine triphosphate, while the ADP is the adenosine diphosphate, they play a major role in the coupling the free energy releasing during the catabolism of fuel molecules to the energy requiring activity of the cells. There is a limited amount of ATP and ADP inside the cell, and the concentration of the ATP is only sufficient to few seconds of energy requiring cell activity. Thus, the ATP acts as a carrier of free energy and not store. The ADP, when it's converted to ATP, there will be it will need about 31 kilojoule per mole to start this reaction, this anabolic reaction. And when the ATP converted to ADP, there will be a release of the energy which is equal to also 31 kilojoule per mole. And we can see that the delta G here is positive because it's an anabolic process, and is need an energy to start, while this there will be a catabolic process and there will be release of the energy. Also, the ADP, when converted to AMP, adenosine monophosphate, this is catabolic process, also release 31 kilojoule per mole. So the phosphate bond inside each molecule act as a preserver of the energy, and because this preserver of energy, these molecules ATP and ADP act as a carrier of energy during the rapid reaction inside the cells. Regardless whether the level of cellular fuel utilization is high with an increased ATP consumption, when the cellular utilization of the ATP high, there will be a consumption of the ATP, or low with decreased ATP consumption, the available ATP within the cell is maintained at a constant level by appropriate increases or decreases in the rate of fuel oxidation. So there is a constant homeostatic process in which the ATP and the ADP stay in a constant state. And when there is a problem in the level of ATP or ADP production or store, and the energy balance, this process or this problems or this deviation from homeostatic process occur in obesity, in hyperthyroidism, or in myocardial infarction. In this process, there will be disturbances in the ATP homeostasis. Now, what is the high and low energy signals? It's high energy signals and low energy signals that inform the cell about its energy state, whether it contains high energy state or it is in a low energy state. So ATP is known as a high energy signal because it signals that the cell has adequate energy level for its immediate need, so it will be stimulate an anabolic reaction. So when there is a high energy in the cell that is labeled by high level of ATP, the cell will convert its process from catabolism to an anabolism. On the other hand, the AMP and the ADP are low energy signals because they signal the opposite and activate the catabolic reaction. When the ATP and when the ADP and AMP is low, sorry, is high, ADP and AMP is high, the cell will understand that the energy status inside the cell is low, so it will convert the processes inside the cell from anabolism to catabolism to release energy during catabolism and to decrease the level of ADP and AMP. Other high energy signal include NADH, NADPH and FAD2H. When these molecules are high in the cell, in their reduced form, that the cell will know that it contains high energy signal and high energy molecule, and the process will be converted to anabolism. While the NADH, NADPH and FAD, which is the oxidized form of this reduced form, when there is an increased level of oxidized form inside the cell will understand that the energy content inside it will be low and will convert its process from the catabolism from the anabolism to catabolism, as we can see in this diagram. The substrate will convert it from its reduced form when there is a high level of high energy substances, which are the substances in its reduced form, stimulate the cell for the oxidative process to release energy low energy molecule and converted from its oxidized form, from its reduced form to its oxidized form. Now, one of these molecules which act as a high energy molecule is the creatine phosphate. The reaction of the creatine phosphate with ADP is reversible in the muscle as in the following process. When there is ATP is high, will react with creatine to produce creatine phosphate, which is a high energy molecule, and release the ADP. And when the cell need a high energy, when the muscle in the exercise, such as in the exercise, need high energy, the creatine phosphate will supply the cell with this energy by its conversion to creatine and release of the ATP to the cell in the process which is facilitated by enzyme creatine phosphate, in which the ADP converted to ATP to be released to the cell during contraction. Creatine phosphate can thus act as small store of free energy in the muscle, in the skeletal muscle or in the cardiac muscle. The store is important in the first few seconds of vigorous muscle activity such as sprinting. Creatine and creatine phosphate both undergo non-enzymatic chemical reaction producing creatinine. The creatine and creatine phosphate in the non-enzymatic not need an enzyme for this reaction to produce creatinine. In this reaction we can see that the creatine converted to creatine phosphate, in which the ATP produced by converting the ADP to ATP, and when the muscle needs an energy source, creatine phosphate will convert it to creatine to release the ATP. Sorry, in this process, creatine converted to creatine phosphate the ATP converted to ADP. When the muscle need the source of energy, creatine phosphate would convert it to creatine, and there is release of ATP. Both of creatine and creatine phosphate converted to creatinine, which is another substance by non-enzymatic reaction, and the creatine will pass to the blood, and it is will be excreted in the kidney to the urine. The rate of production of creatine is proportional to the concentration of creatinine production, or the rate of production of creatinine is proportionate to the concentration of creatine inside the muscle, and this related to the skeletal muscle mass. When there is an increasing in the skeletal muscle mass, there will be an increase in creatine, and so there will be an increase in the level of creatinine. Daily creatinine excretion by male is about 20 to 26 milligram per kg per 24 hour, and is usually greater than that will be excreted by female. This is by male and this is by female because of high muscle mass of the male. An increased excretion of creatinine may indicate there will be an increase in the active muscle wasting, and abnormal high level of creatinine with low urinary creatinine concentration may indicate reduced kidney function. So these are the clinical use of assessment of the creatinine. When the creatinine excretion in the urine is high, that indicates there is a muscle activity or active muscle wasting, and when there is high creatinine in the blood with decreased creatinine excretion by the kidney, that may indicate reduced impaired renal function. And thank you very much.